This chapter starts off with a story. So while I'm reading the story, that's when the late people can show up. So we're going to discuss the opening story, and then we're going to talk about types of vaccines, especially vaccines is a huge thing right now. And then we're going to talk about, will the coronavirus help mRNA and DNA vaccines prove their worth? So from your book, let me make it bigger. If you guys have your book, you can follow along. If you don't, there we go. I'll read it. It's just a couple paragraphs. All right, in February 2003, almost 17 years ago now, thousands of people in Asia became sick from a flu-like disease with high fever, headaches, and respiratory problems. Sound familiar? In the next few months, the disease spread to more than 24 countries across Asia, Europe, North America, and South America. Scientists around the globe mobilized to identify the cause of the illness and quickly isolated a virus called the coronavirus from infected individuals. The disease, named Severe Acute Respiratory Syndrome, which is what SARS stands for, is spread by droplets produced when an infected person sneezes or coughs. Mind you, this story is from 17 years ago. It could be telling the same story today. The current coronavirus is related to the one that came out in 2003. The SARS outbreak was eventually contained by isolating patients and screening travelers for infection. Despite those efforts, the World Health Organization reported that just over 8,000 people were infected with SARS, and about 10% of those died. In the United States, 8 people developed SARS, but all had returned from parts of the world where SARS infections had been reported, and the disease did not spread in the United States. So there's a big difference. By May 2003, scientists using genomic technology determined the DNA sequence of the viral chromosome, which carries 11 genes. We have millions of genes. This virus had 11 genes. To fight future outbreaks, the development of a vaccine was high priority. At that time, no vaccines against human coronavirus had been developed, so researchers decided to try and develop a DNA vaccine. To make a DNA vaccine, one of the virus's genes was isolated using recombinant DNA technology. The viral gene was injected into mice, where it directed the synthesis of a viral protein. The mouse's immune system responded by making antibodies against the protein, which protected the mouse against future infections with the SARS virus. The SARS DNA vaccine was tested on volunteers to see if it was safe and effective. Results from this and other trials are encouraging, but more research and additional clinical trials will be needed before DNA vaccines replace more conventional methods. So conventional vaccines are not made this way, but they're trying everything they can right now for the current coronavirus outbreak. Some concerns about DNA vaccines remain unresolved. Will the viral DNA insert itself into a human chromosome and disrupt the genes that regulate cell growth, perhaps causing cancer? Will the immune system generate an autoimmune response in the volunteer instead of providing protection against the viral protein? Could injecting a viral gene somehow increase susceptibility to the virus rather than prevent infection? Although DNA vaccines are still experimental, using genomic techniques, they can be produced quickly and may be the best defense against viral infections, such as SARS or H1N1. If there's another outbreak of SARS or the H5N1 influenza, which has killed more than 50% of those infected, would you consider using a DNA vaccine as a protection? Would you have your children or other family members treated with a DNA vaccine? So that's a little bit about vaccines. You guys have these websites, so you can look at them if you want, but there's vaccine types from the Health and Human Services website. So normally live attenuated vaccines, when you guys get vaccinated for measles, mumps, smallpox, all that good stuff, it's a live attenuated vaccine. Live vaccines use a weakened or attenuated form of the germ that causes a disease. Because these vaccines are so similar to the natural infection that they help prevent, they create a strong and long-lasting immune response. And that's the point. When you get vaccinated, you want to be protected from that disease for the rest of your life. And then all the way down here in pink, because they're not really a common thing yet, are the DNA vaccines. So the future of vaccines. Scientists are still working to create new types. DNA vaccines are easy and inexpensive to make, and they produce strong long-term immunity. I'm sure there are scientists somewhere working on DNA vaccines for the current coronavirus outbreak, so we can get one and get back to life, back to school. And there's also recombinant vector vaccines, act like a natural infection, so they're especially good at teaching the immune system how to fight germs. So I'm sure there are scientists working on every single type of vaccine right now, but there's a little history on the vaccines for the SARS coronavirus, or for old outbreaks of the SARS coronavirus. And then there's another article, if you guys are interested in reading it, about will the coronavirus help mRNA and DNA vaccines prove their worth? Like I said, they're very experimental right now, but we need all hands on deck to get rid of this virus. So I'm sure there are companies, researchers working on this. All right, so moving on with our notes. You can look further into those. You have the websites if you want. So the carrier of genetic information was DNA. So how did we come to even think that isolating DNA from viruses could help us with vaccinations? So first we got to discover DNA. So nuclei were first purified from cells in the 1860s by a scientist named Friedrich Miescher. The contents of the nuclei contained a substance he called nuclein. Chemical analysis revealed that nuclein was made of SNP, carbon, hydrogen, nitrogen, oxygen, phosphorus. He tested many different types of cells, kidney cells, liver cells, sperm cells, yeast cells, and they were all made of the same chemicals. And then many years later, it was discovered that nuclein contained DNA once we figured out what DNA was. Chromosomes were identified as the carriers of genetic information in the early 1900s. DNA, not protein, is the carrier of the genetic information. Both are present in chromosomes, so they had to isolate each and figure out what was the actual genetic information. Proteins do a lot of work for us, so everybody assumed it was proteins, and they were wrong. Griffiths and later Avery, MacLeod, and McCarty proved that DNA is the transforming factor between bacterial strains. And then Hershey and Chase came a little later and proved that DNA was the genetic material in viruses. We're actually going to watch a couple 2-minute videos about these experiments. So, experiments with mice. This is Griffiths and Avery and MacLeod McCarty.
Do you have the same color eyes as your mom or the same hair color as your dad? Just by observing our appearance, we get a sense that some traits are heritable. They get passed on from parent to offspring. But how does heritable information get transmitted from one generation to the next? In the 1940s, this was a complete mystery. We are going to tell you the story of 3 scientists, Oswald Avery, Colin MacLeod, and Macklin McCarty, who set out to solve this puzzle. Their pioneering research provided proof that deoxyribonucleic acid, or DNA, contains the blueprint of who we are and what we look like. That DNA is in fact the heritable information. This finding may seem obvious now, but back in the 1940s, it was incredibly controversial. Most scientists thought that DNA was too simple to contain all of the information for every single heritable trait, and instead, they thought proteins, which are large and complex molecules, must be the substance passed on from parent to child. At the start of our story, our 3 scientists wanted to follow up on an important finding from a scientist named Frederick Griffith. Griffith worked with a type of bacteria that came in two strains, one that looked smooth and one that looked rough. If he injected the smooth strain into mice, they died. But if he injected the rough strain, the mice lived. To his surprise, if he first killed the smooth bacteria, these dead bacteria did not kill the mice. But if he injected a mixture of dead smooth bacteria and live rough bacteria, the mice died. This meant that amazingly, something from the smooth strain was transferred to and permanently inherited by the rough strain, a process he called transformation. And this caused the rough bacteria to become deadly. This exciting finding left a big puzzle that Avery, McLeod, and McCarty wanted to solve. What was the heritable information that caused the transformation of the rough strain into the deadly smooth bacteria? The scientists purified a chemical substance from the smooth bacteria, and they repeated Griffith's results with it. When they applied the substance to the rough bacteria, those rough bacteria transformed into smooth bacteria. And this trait was heritable and permanent. But the question still remained, what type of molecule was this heritable information? At the time, most people, including Avery, thought the answer was protein. But they found that enzymes that destroy proteins could not block the activity of the substance. It still made rough bacteria look smooth. This meant the substance was not protein. So could it be DNA instead? When they added an enzyme that destroys DNA but does not destroy protein, the transforming activity disappeared. Avery and colleagues confirmed that the heritable information had a chemical signature that matched DNA, and that their preparation had very little contaminating protein or other molecules. The researchers came to the surprising conclusion that DNA was, in fact, the heritable information. Despite the beautiful evidence from these experiments, many scientists questioned whether some contaminating protein might account for the results. It took another experiment by Hershey and Chase in 1952, described in another video, and the discovery of the double helical structure of DNA in 1953 to convince everyone beyond any doubt that DNA is the molecule of heredity. Once people were convinced that DNA was the heritable information, it sparked a wave of new research questions that served as the foundation for biotechnology revolutionizing modern medicine. Many new treatments for human diseases rely on our ability to work with DNA and even pass genetic information from organism to organism. One impactful example is in treating patients with type 1 diabetes who require daily insulin injections. Previously, insulin from livestock was used, but now human insulin DNA is transferred to bacteria, which act as biological factories to produce human insulin for patients. So the early discovery of DNA transformation by Avery, McLeod, and McCarty has had long-lasting benefits for humankind.
So that is our that was the first experiment. The second experiment was by Hershey and Chase. And like I said, I'd rather have videos because they're easier to explain with all of the animations.
Turning the clock back to the 1940s and 50s, the scientific community was in the midst of a debate about the nature of the hereditary material that is responsible for passing our traits on to our children, to our children's children, and so on for generations. Pretty existential question, right? An experiment by Avery, McLeod, and McCarty in 1944 first suggested that proteins were not responsible for heredity, as previously thought, and instead DNA was the culprit. However, given that their results flew in the face of the current thinking, not everyone was convinced. In science, a great deal of evidence is needed before a new scientific idea is accepted as fact. Therefore, in 1952, Alfred Hershey and Martha Chase carried out experiments which addressed a similar question. Was DNA the hereditary material, or was it protein, or was it something else? To solve this important puzzle, Hershey and Chase needed a simple model organism to solve this complex question, and a clever experiment. They chose the simplest organism possible, a bacteriophage, which is a virus that infects bacteria. This virus looks a bit like a lunar landing module and attaches to the outside of the bacteria. It then injects a substance into the bacteria to create many new copies of the virus. These newly manufactured viruses are released when the bacteria bursts. So the substance injected inside of the bacteria must contain the instruction manual for making new virus. It contains the hereditary material. But what was this material that the virus injected into its host? The answer to their question might shed light on the nature of heredity for all life on Earth. Viruses consist of two things, a DNA core surrounded by a protein coat. Hershey and Chase needed to figure out which of the two was injected into the bacteria. They ran 2 experiments, and in each they labeled one of the components. For this labeling, they used radioactive tags, radioactive sulfur for the protein, and radioactive phosphate for the DNA. This was an elegant method because phosphate is only found in DNA and not in the amino acids that make up proteins. And sulfur is only found in proteins and not DNA. After the viruses infected the bacteria, the researchers examined whether the radioactive signal was detected inside the bacteria or left outside. To do so, they needed to separate the bacteria from the viruses attached to their outsides, and did so using a Waring blender, similar to that used for making milkshakes. Once separated, they then centrifuged the sample. Since bacteria are bigger, they were centrifuged to the bottom, while the smaller viruses remained at the top of the tube. When they prepared virus with sulfur-labeled protein and mixed it with bacteria, they found that most of the radioactivity remained with the virus and did not enter the bacteria. When they allowed the virus to replicate and collected the new viruses, less than 1% of the protein radioactivity from the viral parents were found in the newly replicated virus. So the protein was not being inherited. On the other hand, when they performed the same experiment using virus with its DNA labeled with phosphate, they found that most of the radioactive DNA was injected into the bacteria from the attacking virus, and much of this labeled DNA was inherited and passed on to the new virus generation. Hershey and Chase added to the mounting evidence, pointing the finger at DNA as the hereditary material, but they were aware that their experiments left a few unanswered questions, as experiments often do, and they were cautious in their conclusions. From their observations, they stated that DNA had some function in the multiplication of the virus, whereas proteins did not. Word about the Hershey-Chase experiment spread quickly and helped to convince many scientists that DNA indeed was the molecule of heredity, and the remaining skeptics could not hold out for much longer. A year later, Watson and Crick published their model of the DNA double helix, which provided an elegant explanation for how DNA with such a simple structure could be replicated and pass along information from parents to offspring. The Hershey-Chase experiment is considered a classic in biology because it tackled an important question with the use of a well-chosen model organism and an elegant experimental design in order to clearly distinguish between 2 competing hypotheses.
So that brings us to Watson and Crick, which is what everybody hears when they think of DNA. So a review of chemical bonds really quick. You have covalent bonds. Covalent bonds are represented by a line. You have double covalent bonds represented by 2 lines. And then you have hydrogen bonds. So in between the H's and the O's of water molecules, you have hydrogen bonds. Hydrogen bonds are fairly weak and broken very easily. But the chemistry of DNA are nucleotides. This you learn, I know a lot of you learned this in my class anyways. DNA is made of nucleotides. Nucleotides have 3 parts. They have a nitrogen-containing base. There's 2 categories. There's purines and pyrimidines. Purines are A and G, adenine and guanine, have a double ring structure. You can see in the picture, it looks like they're 2 rings stuck together. And then pyrimidines, C and T, cytosine and thymine, have a single ring structure. Just looks like 1 ring. And then uracil we'll talk about in a little bit when we get to RNA. The second and third component, the second we have sugar. Sugar is deoxyribose in DNA, and then ribose in RNA. They're both 5-sided. They look like a home plate in baseball. And then there's a phosphate group. The phosphate group is the same for both. Nucleotides are joined by covalent bonds between the sugar and phosphate groups to form polynucleotide chains. So you can see these, the S is sugar, the P is phosphate, and they build chains all the way up. And covalent bonds stick the sugar and the phosphate together. And then in the middle, where the base pairs are, these are connected by hydrogen bonds. So you have a lot of different bonds in this molecule. Yes, Shay, we'll talk about that at the end. We got a little blurb about RNA in the end. All right, determining the structure of DNA. This is where everybody starts. Everybody starts with Watson, Crick, Wilson's, Rosalind Franklin. This is Rosalind Franklin's famous picture that Maurice Wilkins actually stole and gave to Watson and Crick. And Watson and Crick made it the basis of their research without giving credit to Rosalind Franklin. But your ed puzzle this week is about this situation, so I won't go over it too much. But these are the 4 scientists that you usually hear about. You usually don't hear about Hershey and Chase and Avery and MacLeod McCarty and Friedrichs. But there were scientists leading up to Watson and Crick. Watson and Crick didn't just pull this out of thin air. They built on the scientists that came before them. But from their model, the following was discovered. DNA is composed of 2 polynucleotide chains. It's a ladder-like substance. The sugars and phosphates form the side rail. So if you're climbing that ladder, you're grabbing onto the sugar and phosphate on the side. And then the rungs, the part that you step on, those are all the nitrogen bases. Like I said, sugar and phosphate have covalent bonds. And bases are held together by hydrogen bonds. And the chain runs in opposite directions. They're called anti-parallel. And then they're coiled, twisted around to form a double helix. So the ends, you can see in the picture. And go ahead to do this. On one side, you start at the 5' end. The little apostrophe right here is called prime. So we call this the 5' end. And then it goes down here to the 3' end. And then the other strand is the opposite. At the top, you have the 3' end. It goes all the way down to the 5' end. So because they run backwards, they're called anti-parallel. And that's what the ends are called. Also from their model, they discovered base pairing is highly specific, which Erwin Chargaff actually took Watson and Crick's model and made up these rules. They're called Chargaff's rules. The A in one chain always pairs with the T in the opposite chain, and the C always pairs with the G. If you have trouble remembering that, just remember A and T are straight letters. You have a T and an I, and they're made of sticks. They stick together, T and an A. And then C and G are the curly letters. You got a C and you got a G, and the curly letters stick together. And between the A and the T, there are 2 hydrogen bonds that link it. Between the C and the G, there are actually 3. The predictable base pairing makes the 2 chains of DNA complementary. So if you have a list of letters and I ask you for the complementary letters, you just tell me whatever the opposite is that they pair with. T pairs with A pairs with T, G and C go together. So if one strand has 5' A, C, G, T, C to the 3', the other strand starts at the opposite, starts with the 3' end, and the A pairs with the T. I'll do this. There we go. The A pairs with the T, the C pairs with the G. That G will pair with the C. That T goes with the A, and the C goes with the G. So you will do this quite a bit in your quiz, your practice questions, and your test. RNA, a second type of nucleic acid, is found on both the nucleus and cytoplasm of cells. Its function transfers genetic information from the nucleus to the cytoplasm, participates in the synthesis of proteins. It's a component of ribosomes. It participates in the regulation of gene expression. It has the same general structure. Nucleotides are joined by covalent bonds to form polynucleotide chains, but there are some differences. One is that RNA is single-stranded. It'll fold upon itself when needed, but it doesn't have that second strand to make that double helix structure. So it would be like climbing up half a ladder. Nucleotide building blocks, there's some other differences here. You still have the purines and pyrimidines, but the pyrimidines this time, you have a C and a U. There's no thymine, there's no T's anymore. Like Shay said, she put in the chat, the thymine is replaced by the uracil. The sugar is also a little different. It's ribose instead of deoxyribose. That's where the R in RNA comes from. And there's still a phosphate group. And then you can see in the chain, you have the sugar, phosphate, the sugar, the phosphate, the sugar, the phosphate. Makes a chain all the way up or all the way down, depending on how you look at it. DNA has 2 major functions in the cell. It'll replicate itself and to carry genetic information for proteins and RNA molecules necessary for life. It has to replicate itself so that when our cells go through meiosis, our cells will have the full amount of information that it needs to function. Before a cell divides, the DNA must be copied, so each daughter cell will receive an exact copy of the genetic information in the chromosomes. I don't know what just happened. It occurs during S phase of the cell cycle. We talked about the cell cycle. G1 for growth, S for synthesis. This is DNA synthesis when it copies. G2 for more growth, and then M for mitosis when the cell splits. Replication begins at specific sites on a double helix. They're called the origin of replication. It proceeds in both directions. So the DNA is double stranded. Its strands split apart, and both strands get copied. One side replicates continuously. The other side has to replicate backwards, so it does it in fragments. Those fragments are called Okazaki fragments, after the scientist who discovered them. So you can see in the GIF, the top strand, when it splits, the top strand just goes along nice and smooth. It gets copied as the DNA strand opens itself up. The bottom strand goes the opposite way and gets put together in fragments. Stages of replication. Enzymes will unzip, unwind the double helix to make a replication bubble with a fork where it splits. The enzyme that splits it is called helicase. And then each strand acts as a template or a hard copy. There's a couple different animations of how that happens. Bases that are floating free in the nucleus bond to their mates on the original DNA strand. The enzyme that directs this process is called DNA polymerase. So for example, if the parent strand has a T, the daughter strand is going to have what? What pairs with the T? You guys put it in the chat, or someone put it in the chat. Or make sure you know in your notes. If the T pops up in the parent strand, then a specific one is going to pair with it in the daughter strand. And then enzymes bind the two new molecules of DNA together. This enzyme is called DNA ligase. It's like a glue. Each new DNA has one old strand and one new strand in its double helix. And it's called a semi-conservative process because it conserves half that information. So our bodies don't have to make it new all the time. We just use what we have to make it half new. The three-dimensional structure of chromosomes is important because we have about 2 meters of DNA, but it has to fit in our cells and our nucleus. Our nucleus only has a 5 micrometer diameter. We have a lot of information to fit in a very, very tiny space. But chromosomes uncoil and recoil during the cell cycle. And the spatial arrangement of DNA is important for the regulation of gene expression. We looked a little bit at epigenetics a couple chapters ago and how DNA can be really tightly coiled. If it's really tightly coiled, those genes aren't going to be expressed. But if it's uncoiled and loose, that means enzymes can get to it and replicate it and express that DNA. Within chromosomes, DNA is coiled around in clusters of histones to form nucleosomes. So you can see in the picture, it starts as DNA on the bottom, and then it wraps around histones. The individual proteins are called histones. And then if you put it all together, the DNA in the histone is called a nucleosome. And then that wraps around itself, and it just keeps twisting and twisting and twisting until you get the chromosome structure. And that 2 meters of DNA fits inside the 5 micrometer diameter of your nucleus, of the chromosome. The genome can fit inside a nucleus due to extensive coiling, of course. That's what an actual chromosome looks like under, I think it's an electron microscope, a scanning electron microscope. And specific regions of chromosomes have important roles. We've touched on it before, the centromeres and the telomere. The centromere is that central region. The telomere are the ends. During cell division, the DNA in centromeres serves as a platform for the assembly of proteins that form a plate. That plate is called the kinetochore. The spindle fiber is attached to kinetochores during metaphase and move chromosomes apart during anaphase. So if you remember mitosis, those chromosomes, they come together, they line up in the middle, and then they get ripped in half. And they get ripped in half in that centromere region because kinetochores have developed with spindle fibers, and the spindle fibers are the actual strings that pull them apart. Telomeres are short DNA sequences repeated hundreds or thousands of times, located at the end of each chromosome. The chromosome looks like the little X. It's got four little ends, 4 little telomeres. The human telomere sequence. We have a specific sequence at the end of our chromosomes. It's TTAGGG. Because of the way DNA replication takes place, each round of replication leaves some telomere repeats uncopied, making chromosomes a little shorter after each round of replication. Our mechanism gets a little bit lazier each time and leaves a little bit of our chromosomes uncopied to be able to pass on to the next set of cells. In most cells of the body, telomeres shorten with each division, meaning that cells can divide only a certain number of times before they stop being able to divide. Telomere shortening may be linked to the aging process, but whether it's the cause or effect is still debated. So scientists are also always looking at anti-aging, how to keep us younger, how to help us live longer. Right now, they're seriously looking at telomeres in that process. And some cells, such as those forming sperm and eggs, contain an enzyme called telomerase, which makes extra telomere repeats, keeping the number of telomeric repeats constant from one cell division to another. So not all of our cells shorten our chromosomes every time. Sperm and eggs, of course, which is important for our reproduction, have a way to prevent this. So telomerase is active in more than 90% of cancers, allowing cancer cells to divide indefinitely. The definition of cancer is uncontrolled cell division. So because telomerase is not active in most cells, but it is active in cancer cells, the development of a telomerase inhibitor may prove to be an effective treatment for cancer. Cancer cells tend to have a lot of this enzyme. If we can block that enzyme, the cancer cells would hopefully shorten in each cell division and eventually go away altogether. That's the goal, to get rid of cancer, get rid of tumor cells. And then chromosome territories. In the nucleus, each chromosome occupies a distinct territory separated from other chromosomes by a region called the interchromosome domain. The territories are linked to functions, but they're not fixed. They move around in the nucleus at different times of the cell cycle. It's like liquid or jelly in there. It's not solid, so they kind of move against each other and switch around. So it's hard to pin down a function because they move around so much. And that is it for the notes today. You guys have any questions? You're gonna have your Rosalind Franklin Ed puzzle, and then your discussion this week. We did racism in science, so now we're gonna do sexism in science. You're gonna research a female scientist that didn't get credit for the work she should have, or the work that she discovered. Like I said, any questions? Give me a yes or no in the chat box. And let me know. You guys are awful quiet today. Yeah. All right. If you guys don't have any questions, feel free to go and start working on one of the assignments.
Bye, Ms. Reese. Take care.
Bye. Take care. Have a good day. Have a good day, Julian. Have a good day, Jasmine.